Our lecture today will deal with the ventricles. The ventricles are spaces located deep inside the brain, which contain CSF. Lateral ventricle. The paired lateral ventricle are located on either side of the brain. The lateral ventricles contain the choroid plexus, which produce CSF. So, the choroid plexus produce CSF. Note, the choroid plexus is almost always calcified in adults, as shown here in this picture. Look to the arrow, white arrow. The third ventricle, this is also a CT for the brain. The third ventricle is located centrally. The lateral ventricles communicate with the third ventricle via small hole. This is the foramen of Monro. And the white one arrow show the foramen of Monro that connect the lateral ventricle with the third ventricle in this picture. The fourth ventricle. The fourth ventricle is located in the posterior fossa between the brain stem and cerebellum. It communicates with the third ventricle above via a very narrow canal, the aqueduct of Sylvius. The basal cisternae, that CSF in the basal cisternae, surrounds the brainstem structures. Brain parenchyma and loops. The brain consists of gray and white matter, as you know, structures, which are differentiated on CT by differences in density. The white matter has a high content of myelinated axons. Gray matter contains relatively few axons and a higher number of cell bodies. As myelin is a fatty substance, it is of relatively low density compared to the cellular gray matter. White matter, therefore, appears blacker than gray matter. So, should concentrate on this point that the gray matter appears gray, white matter appears blacker. See here in this slide, gray matter, the white matter is located centrally and appears blacker than the gray matter due to its relative low density. Clinical significance of this, that pathological approaches may increase or decrease the differentiation in density between gray and white matter. So we can benefit from this point. Brain loops. The brain has paired bilateral anatomical areas or loops. These do not exactly correlate with the overlying bones of the same names. So the brain loops that in the CT brain, that is superior part, superior slice on both sides, the frontal loop are separated from the parietal loop by the central sulcus, that is the arrowhead, the white arrowhead, central sulcus. Note that the frontal loop are larger and the parietal loop and occipital loops are relatively small. Also, the brain loops, as you seen here in the CT brain, inferior slice, the most anterior part of the frontal loop occupy the anterior cranial fossa, this is the green one. The temporal loops occupy the middle cranial fossa, that is the BMB one, and the cerebellum and brainstem occupy the posterior fossa, that is the blue and the red one, as seen in this CT. Now, cerebral vascular tertiaries. Different areas of the brain are supplied by the anterior, middle, and posterior cerebral arteries, as when it was discussed in the previous lecture, in a predictable distribution. The posterior fossa structures are supplied by the vertebrobasilar arteries. The arteries of the brain are not well visualized on conventional CT, but knowledge of the areas of the brain they supply is helpful in determining the source of a vascular insult. Insult, that is insufficiency. So, the key points of this slide that the cerebral and vertebrobasilar arteries supply regions of the brain in predictable distribution. Gray matter structure. Important gray matter structures visible on CT image of the brain include the cortex, insula, basal ganglia, and the thalamus. See on this slide, each one. The cortical gray matter, the gray matter of the cerebral cortex is formed in folds called gyri. Note that the cortex appears whiter, denser than the underlying white matter. Insula. The insula forms an inner surface. This is the green, this blue area of the cerebral cortex, found deep to the cerebellum fissure. Note that the cerebellum fissure, and it is deep to it. The insula clinically significant that loss of definition of the insular cortex may be an early sign of acute infarction involving the middle cerebral artery, tertiary. So it is very important that when we find that there is loss of definition of insula, it means that there is some disturbance in the middle cerebral artery, infarction or thrombosis or same like this. Now, basal ganglion and thalamus. The thalamus and the basal ganglion are relatively identical with CT. Basal ganglia include the lentiform nucleus, caudate nucleus. Basal ganglion clinical significance. Insult to the basal ganglion may result in disorders of movement. So, any lesion in basal ganglion will lead to disorders of movement. Thalamus clinical significance. Insults to the thalamus may result in thalamic pain syndrome. White matter structures. White matter of the brain lies deep to the cortical gray matter, as you know. The internal capsule are white matter tracts which connect the corona radiata and white matter of the cerebral hemisphere superiorly with the brainstem inferiorly. The corpus callosum is a white matter tract located in the midline. It arcs over the lateral ventricle and connect the white matter of the left and right cerebral hemisphere. What is the key points of this slide? The internal capsule and corpus callosum are clinically important white matter tract. So the types of tract, we have a commissural fibers that composed of commissural fibers allow communication between cerebral hemispheres like corpus callosum, which is the largest commissure. Associated fibers connect different parts of the same hemisphere. Projecting fibers runs vertically, descend from the cerebral cortex or ascend to the cortex from the lower regions in the spinal cord. Slide showing the associated fibers and showing the corpus callosum, the largest commissural fiber. Here in this slide, we can see that there is projecting tracts, descend or ascend, and they decussate in the pyramids. And we can see here also the commissural tract that connect the right with the left side or hemisphere. Internal capsule. The internal capsule are narrow white matter tract which contain a high number of axons connecting the corona radiata and cerebral hemisphere white matter superiorly to the brain stem inferiorly. Each internal capsule, it has an anterior limb and a posterior limb connected to the of the genie. Genie is means that this like the region between the anterior limb and the posterior limb, as seen here in the CT scan. Internal capsule clinical significance. What is? The internal capsule are supplied by perforating branches of middle cerebral arteries. As these vessels are small, they are susceptible to infarction. Even a small insufficiency to the internal capsule can have a profound effect on the motor and sensory function in this region. The corpus callosum. Corpus callosum, that is CT brain, CT image. This is the blue one. Sagittal CT image shows the corpus callosum as a midline structure arching from anterior to posterior. Posterior fossa. The posterior fossa accommodate, as you know, the cerebellum and the brainstem. Superiorly, the cerebellum is separated from the cerebral hemisphere by tentorium cerebelli. These all informations we take it in two last lectures previously. Posterior fossa, the brainstem and cerebellum occupy the posterior fossa here. This the pons and cerebellum, as you see in this CT. Posterior fossa also, imaging software allows visualizations of the structures of the brain in different planes. The sagittal plane can be useful to show the anatomy of the brainstem. Note that the MRI may be required if there is a specific concern regarding the brainstem pathology. So MRI is benefit in if the lesion is present in the brainstem. See here the brainstem, midbrain, medulla oblongata. This is the blue structure in this slide or CT. Now, vascular tertiaries. The vascular tertiaries above lateral ventricle, the anterior cerebral arteries supply a narrow band of the cerebral hemisphere adjacent to the midline. Anterior. This is the blue one. The middle cerebral artery supplies the largest area of the brain. This is the red one. While the yellow one is the posterior cerebral artery as seen here in this CT. Calcificate structures. There are several structures in the brain which are considered normal if calcified. Knowledge of these structures helps avoid confusion, especially when considering if there is an intracranial hemorrhage present. The commonly calcified structures, we should know it, include the choroid plexus, pineal gland, basal ganglia, and falx. The key points of this slide: the commonly calcified structure of the brain include the choroid plexus, as we say, pineal gland, basal ganglion, falx. Use of CT that is helpful in differentiating calcified structures from acute hemorrhage. So here in this scan, we can see calcified choroid plexus. In adults, the choroid plexus of the lateral ventricles is almost always calcified. See it. It is in the red circle here in this CT. Here, a calcification of the pineal gland. The pineal gland is located immediately posterior to the third ventricle. It is very commonly partly or fully calcified in the adults. Look, this calcification, white one, the third ventricle is the yellow, while the pineal gland is the green one. Also, basal ganglion. Calcification of the basal ganglion is commonly in elderly patients. This is also shown in the red circle here. Falx, calcification of the falx cerebri. The falx is commonly calcified in adults if viewed on the brain windows only. Calcification of the falx can be mistaken for acute intracranial blood. Please don't mistake from this point. Use of the CT show calcification of the falx are more clearly. This is shown as a white thing. This is in the CT. This by brain windows, we can see this calcification. Now, in the normal anatomy, we can see also the different structure like the orbit, like the sphenoid sinus, temporal loop, external auditory canal. This is all can be seen by this CT scan. Or here also in this slide, we can see that frontal loop, frontal bone, dorsum celli, basilar artery, all these structures can, mastoid air cells can be clearly seen by. Also here, Sylvius feature, temporal loop, frontal loop, supracellular cistern, midbrain, fourth ventricle, all shown here by the CT scan. Here also we have a sagittal image, and we see that different parts like the pons, midbrain, corpus callosum, all these structures can clearly be shown in the CT scan.